Hello friends, welcome to Biology Extrema, where we teach biology from the basics with extreme. Myself Keshav Bansal, and today we are going to study charging of tRNA, okay, or we can say aminoacylation. Clear? So first relate this topic, from which topic this subtopic comes from. So it is a part of molecular biology, okay. It is a part of molecular biology. What happens? We all know that from DNA, okay, DNA, RNA is formed, and specifically mRNA is formed with the process of transcription, okay. With transcription, mRNA is formed, and from this mRNA, what happens? Through translation, proteins are formed, okay. This way DNA expresses itself. Clear? So this is translation, okay. So what is a protein? Protein is nothing but a chain of amino acids. It's a polypeptide chain, okay, in which amino acids are connected by peptide bonds. So these are peptide bonds, okay, and these beads are what? These are amino acids, okay. These are amino acids. So we know that proteins are made up of amino acids, okay. Now we also know, if we have studied the translation process, that we need three major components. We need a lot of other factors too, but basically major components are we need ribosomal RNA, okay, which will bring the which are present in the ribosomes, okay, which will provide the place where which will provide a platform where all this reaction will occur, okay. We can say the ribosomes. We will require a mRNA, okay, which will bring the codons, the information of which kind of amino acids to attach, okay, to form a particular type of proteins. Clear? And we will bring a tRNA, whose job is to bring the amino acids itself, okay, who will carry the amino acids. So what happens? Without this tRNA, without this part, the protein synthesis will not occur, okay. So we know that tRNA will bring the amino acids, and it will keep attaching to the amino acids and make a protein. We bind tRNA to the amino acid, and the binding of the tRNA to the amino acid, this process is called, we can say, charging of tRNA, okay, charging of tRNA, or we can say activation of tRNA, or we can say it aminoacylation, okay. We can say it aminoacylation. Clear? Now we'll study it in a bit detail, okay. So we know that attachment of an amino acid to the tRNA is called charging of tRNA, or we can say activation of tRNA, okay. And we can also call it aminoacylation, okay. Why we say that? Because when we charge an amino acid, a tRNA molecule with the amino acid, okay, we now the tRNA, first it was a tRNA, first we call it a tRNA. Now the tRNA, after charging, it is called aminoacyl tRNA, okay. It is called aminoacyl tRNA, and the tRNA is, we can say, it is charged acylated, okay, or the process is called aminoacylation. So charging, activation, or aminoacylation, it's the same thing. Clear? And this reaction is catalyzed by an enzyme, okay. This enzyme can be amino, because we are forming aminoacyl tRNA, okay. So we can say aminoacyl tRNA synthetase, okay. And we also know that we are ligating the amino acid, okay, to the tRNA molecules. So we can say aminoacyl tRNA ligase. So this is the enzyme, okay. This is the enzyme aminoacyl tRNA or aminoacyl ligase. Clear? Which helps in this phenomena. Clear? Now if I ask, I can ask two things, that how many types of this aminoacyl tRNA synthetase, we will now use the word enzyme, okay, to refer the that particular enzyme. How many types are there, okay, and how many times I have to use that if I am doing a process of translation, okay. So let's first cover this times, okay. If I am forming a protein with 6 amino acids in it. Okay, so I have to use this enzyme 6 times. Okay, so the number of amino acids, okay, these amino acids can be same, they can be different. Okay, and also make sure that note that the enzymes are specific to amino acids. Okay, so one enzyme means one amino acid, one specific amino acid. Okay, the enzyme going to be different for the for a particular amino acid. Clear? And so the number number of amino acids I use to make a protein, so the times I use the enzyme is the same. Clear? So it's the number of amino acids I use. Clear? Now these amino acids can be same. Okay, as I told you, these amino acids can be same, so the enzymes can also be same. Okay, it can be same, it can be different if the amino acids used are different. Okay, for example, here we used leucine, we used tyrosine, we used leucine. Okay, we used guanine. So these two enzymes will be same. Okay, but this will be different and this will be different. Clear? Getting my point? Now if I talk about the types of enzymes there, okay, like this belongs to one type of enzyme. Okay, this belongs to another type of enzyme, this belongs to another one. Okay, so how many types of enzymes are there? So it depends on the type of amino acids. Okay, so the amino acids, we know that there are 20 amino acids. Okay, 20 protein synthesizing amino acids which are used in protein synthesis. Okay, now we also know that they are 21st and 22nd amino acids, solanocysteine and pyrrolysine. Okay, but still some textbook use 20 for the amino acid, that are 20 protein synthesizing amino acids. Okay, so the number of protein synthesizing amino acids determine the number of determine the types of enzyme. Clear? So this is the case. If there's sometimes they're written 20, that will be correct, and if it is even written 22, then it is also correct. Okay, so now we will talk about that how this enzyme did this phenomena. Okay, how it attaches the amino acid to the tRNA. Let's talk about it. So it does it in 2 steps. First is adenylation and second is tRNA charging. Okay, first one is adenylation and second is tRNA charging. So what happens, as we can see in the figure, that this is our enzyme. Okay, these are enzyme. So what happens, first an amino acid came, this amino acid came, and what another molecule came, it is called ATP, adenosine triphosphate molecule. Okay, tri means it has 3 phosphate molecule. Clear? So amino acid is bound to the ATP molecule with the help of this enzyme. Okay, and when what happens during the binding from the ATP, first there was ATP molecule and the amino acid. Okay, now they get attached to the on the sides of the enzyme. Okay, to the sides they get attached, and the enzyme, what it does, it binds the amino acid to the ATP. During the binding, what happens, 2 phosphate molecules from the ATP get removed. Okay, when we are using these 2 phosphate, we are referring them as pyrophosphate. Okay, 2 phosphate molecules which are removed are called pyrophosphate. So from adenosine triphosphate I remove pyrophosphate, and what I get, adenosine monophosphate. Okay, because there is just one phosphate group attached to it. So during the binding, 2 pyrophosphate is removed, and as a result ATP is now bonded to the AMP. Okay, the amino acids are now bounded to the AMP, adenosine monophosphate. Clear? Now this is the adenylation part. Okay, amino acid reacts to the ATP and AMP is transferred to the amino acid. Okay, pyrophosphate is removed. You can also add it here. Now what happens? Now the tRNA molecule will came. Okay, the tRNA molecule will came and it will replace the AMP. Okay, so the AMP will be released, and now the tRNA is bound to the amino acid, and after this final product is found, the enzyme will release this product. Okay, so this part is tRNA charging, the transfer of AMP linked amino acid First it was linked, after adenylation it was linked to AMP, amino acid. Now it is bind to the tRNA, okay, and AMP is released, and to tRNA we bound it to 3' end, because we know the structure that this is the 5' end, okay, and this is the DHU loop, this is the anticodon loop, this is the pseudouridine loop, and this is the 3' end, and on 3' end there is CCA, terminal codon, okay. To this point the amino acids are attached, okay, to this point the amino acids are attached. Now this enzyme amino acid tRNA synthetase is categorized in two classes, okay, the class 1 and class 2. Based on what it is categorized, what happens, that on the terminal nucleotide of the 3' end, okay, I just draw the tRNA a moment ago, that to 3' end there will be a terminal nucleotide attached to it, okay, and to that 3' end, okay, to that nucleotide at the 3' end of the tRNA, to the terminal, which is the last one, okay, it has either 2' OH group, okay, this is the part of tRNA, okay, and either 3' OH group. And if I add amino acid using the enzyme to the 2' OH group, okay, then the enzyme comes in class 1 category. I'm repeating again, the tRNA molecule, we know that it has 5' and a 3' end, okay, and to 3' end that terminal nucleotide present, okay, it is made up of nucleotide sequence, okay. So at the terminal nucleotide at 3' end of the tRNA, it has 2' OH group and 3' OH group, okay. If I add amino acid to the 2' OH group of the terminal nucleotide, okay, then the enzyme is categorized in class 1 category, okay, and if I attach it to the class 2 enzyme, then to the 3' OH group, then the enzyme is categorized as class 2 category. Clear? So on this basis, to which group we are adding, it is categorized, okay, and the class 1 enzymes are monomers, while the class 2 enzymes are dimers. So this is all about the charging of tRNA. I hope you like the video, okay. If you like the video, please subscribe our channel and share these videos, and if you can donate a small amount, so we can keep continuing doing this noble work of providing free lectures and quality notes. Thank you from Biology Extrema. See you next time. Take care.